Namaste students, Hamasha Khushro Vidya. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth Women's University, Jaipur. So today we are here to discuss your practical session, the TS of Testis. Okay, so this comes under the subject Developmental Biology Lab, and this was incomplete in the previous lecture. So let's repeat the session and go ahead. So for the study of TS of testis, we have a basic idea about this organ. So this is the primary reproductive organ of a male. Primary reproductive organ means it will produce the gametes. Okay, for performing this experiment, you need two things. One is the TS of testis, either mice or rabbit or rat or human. So you need a compound microscope. Digital microscope will be better, so that you can take the images, and you need a tissue paper. So you for wiping the lenses, slides, etc. So the procedure: first of all, you will clean the slide and lenses of the microscope. Then you fix the slide on the stage of the microscope, and you will focus and observe the anatomy of the testis. So you will visualize the things, and you can relate whatever you study in the theory session. So in the TS of the testis is found in a pair in the males. Okay, so in the anatomy of the structure, it comprises several seminiferous tubules that embedded in the interstitial tissues. So the seminiferous tubules are the place where the gametogenesis takes place, and a thick fibrous tissue called tunica albuginea covered the testis, and it comprised different types of cells from the outside to the lunar in the manner given here: the spermatogonia, then spermatocytes, then spermatids, and finally the spermatozoa or the sperms. So the Sertoli cells are located between the germinal cells and some other cells. The leading cells are also there that produce the testosterone and are present in the interstitial tissues. So the three phases are there: multiplication phase, growth phase, and maturation phase in the spermatogenesis process. Okay, so this is the whole anatomy of the testis, and this is the vasa deferens. Okay, the body of the epididymis, the head of the epididymis, the tail of the epididymis. These are the seminiferous tubules, and the covering is tunica albuginea. Okay, so the testis is on the base portion. It is surrounded by the uh, two layers of the tunica vaginalis, and the space between these two layers is known as the vaginalis cavity. Okay, here these are the tunica vaginalis, and this is the tunica vaginalis cavity. The tunica albuginea layer is there, and these chambers are there, and these are the seminiferous tubules. They combine here as a retitestis and leaves this ovular structure as a vasa efferentia, and this is the epididymis, and this is vasa deferens. Okay, so the precautions you need, that you should clean first the microscopic stage, the lenses, the slide, and you focus optimally. Okay, so for observing into the microscope, the focus is the most important thing. So patiently you will focus at the various anatomical structures on the testis, and you will correlate with the anatomy you have studied. So the microscope should be handled with care, because it is a sophisticated and a delicate instrument, and adjust the lenses such that the focus is better, and observe the images, take the images with the digital microscope, and put it as a result, and observe. And thank you. So hope you are satisfied with this digital session, and if you have any query, please write in the comment box and I will resolve your query. So this digital session is powered by digital version 2, Jyoti Vidhyapeeth Women's University, Jaipur. Thank you.